So without further ado, I'd like to introduce the speakers in our first session. Kim Smith will begin with an overview of FDA, its structure and mandate. Jim Smith will cover the basics of clinical trial design, and Mark Levinson will review the statistical principles for drug development. So Kim, over to you.
Thank you for that introduction. For the next 20 minutes, I'm going to provide you with an overview of the FDA structure and mandate to provide some background for later topics that we'll cover during the course. So the objectives of my talk are to provide a brief history of the FDA, to describe the current mission of FDA, and to introduce FDA's legal and regulatory framework for drugs and biologics. So to start with a brief history of the FDA, I think it's important to recognize that the agency was built on a legacy of public health failures, so unsafe, ineffective, counterfeit, or adulterated drugs, as well as some unethical practices and fraud. The image on the right is an advertisement for Mrs. Winslow's soothing syrup, which was marketed in North America and the United Kingdom in the 19th and early 20th centuries as a cure-all medicine for fussy babies. But the primary ingredients were morphine and alcohol, and thousands of children are believed to have died from overdoses or from morphine addiction and withdrawal. The lower right is an image from the New York Times. This is on the Tuskegee syphilis study, which was started in 1932 by the U.S. Public Health Service working with the Tuskegee Institute. It was a study to record the natural history of syphilis. Participants were not consented and were told that they were being treated for bad blood. By 1943, penicillin was the treatment of choice for syphilis and was widely available, but the participants were never told about this or offered treatment. In 1972, the study was exposed when an article was published by the Associated Press, but unfortunately at that time, men in the study had been untreated for many years and their disease had progressed. To continue with our brief history, I'll provide an overview of just some key milestones in U.S. food and drug law. So starting with 1848, we had the passage of the Drug Importation Act, which required U.S. Customs Services to inspect entry ports for the entry of adulterated drugs from overseas. It's really one of the earliest attempts to regulate the U.S. drug supply. Moving forward to the early 1900s, at that time, diphtheria patients were routinely treated with an antitoxin that was derived from the serum of horses. The horse serum was manufactured locally and there were no central or uniform controls. In 1901 in St. Louis, Missouri, the blood of a tetanus-infected horse was used to develop the antitoxin and was administered to 13 children who ended up dying of tetanus. This tragedy led to the 1902 passage of the Biologics Control Act, which allowed for the first controls over processes used to produce biological products, including vaccines, serum, and antitoxins. In 1906, we had passage of the Pure Food and Drug Act, which prohibited interstate commerce in misbranded and adulterated foods, drinks, and drugs. In 1912, we had passage of the Shirley Amendment. The Shirley Amendment prohibited labeling medicines with false therapeutic claims intended to defraud the purchaser, but it was a standard that was very difficult to prove. Moving forward to the late 1930s, in 1937, we had another tragedy. At the time, sulfanilamide was a drug that was used to treat streptococcal infections, and there was demand to provide it in a liquid form. So a company experimented and found that sulfanilamide dissolved in diethylene glycol, compounded the product, and sent it all over the country. Unfortunately, diethylene glycol is antifreeze and is toxic. The product killed 107 people, many of whom were children, and really provided a dramatic example of the need to establish safety before marketing a product. This tragedy led to the passage of the 1938 Food, Drug, and Cosmetic Act. The act required that new drugs be shown to be safe before marketing and really started a whole new system of drug regulation, and it was the origins of much of what we do today. In 1944, we had the Public Health Service Act. The Public Health Service Act covered a broad spectrum of health concerns and included provisions related to the regulation of biological products. In 1951, we had the Durham-Humphrey Amendments. Durham-Humphrey Amendments defined the kinds of drugs that could not be used safely without medical supervision and restricted their sale to prescriptions by licensed practitioners. So this was our first foray into prescription products. We then have our next tragedy. thalidomide in the 1950s and early 1960s was widely used to treat nausea in pregnant women. It became apparent in the 1960s that thalidomide was resulting in severe birth defects in thousands of children, limb deficiencies that you can see in the images on the screen. This was primarily occurring in Western Europe, and fortunately, the product had never been approved in the United States. Dr. Francis Kelsey, who was a FDA medical officer, had concerns about the amount of safety data that was available and had kept the drug off of the US market. This example provided a lot of public support for stronger drug regulation and led to the 1962 passage of the Kefauver-Harris drug amendments. These amendments ensured drug efficacy and also enhanced safety controls. For the first time, drug manufacturers were required to prove to FDA that their product was effective before the product was marketed. In 1968, we had the Drug Efficacy Study Implementation, or DESI. Basically, many products had been approved between 1938 and 1962 on the basis of safety alone, and the agency had to generate a process to go back and review 4,000 plus drugs to determine whether they should stay on the market. The products that did stay on the market went through this DESI approval process. So fast forwarding a bit to the FDA now, the agency has over 18,000 employees, many of whom are scientists. Recent numbers are that over 11,000 of these 18,000 employees are in science fields. The agency oversees the safety of more than $3.6 trillion worth of food, tobacco, and medical products, accounting for 21 cents of every dollar spent in the US. On the right, you see a figure that was from 2023, although remains largely current. It shows that the human drugs program is the largest by far, followed by foods, tobacco, devices and radiological health, and then biologics. This course will really focus in on the human drugs and biologics programs. So I'm going to shift focus a bit to provide some information about the legal framework that guides the work that we do at FDA. Our work is grounded in statute with the Federal Food, Drug, and Cosmetic Act and Public Health Service Act providing our statutory authority for oversight of clinical investigations to evaluate safety and effectiveness. The laws are further explained through regulations that the agency issues. They can be found in the Code of Federal Regulations. This is how we implement the statutory authority over the conduct of clinical investigations. A few key sections. Title 21 is Food and Drugs, and Chapter 1 addresses the Food and Drug Administration. Subchapter A provides some general provisions. This is where you'll find information, for example, on informed consent and institutional review boards. Subchapter D addresses drugs for human use, and Subchapter F addresses biologics. FDA guidance is also issued regularly by the agency, and FDA guidance is intended to be advisory. It's intended to assist regulated entities in complying with the regulations and to help them to understand FDA's current thinking on a topic. There are numerous guidances, many of which are relevant to investigators, others might be more relevant to sponsors, and if you're interested in whether there's guidance on a particular topic, the link at the bottom provides the search location. So much of the agency's work revolves around review of submissions to certain types of applications. The first is the investigational new drug application, or IND, and then you have the new drug applications, NDA, or biologics license application, or BLA. And I'll talk a little bit more about each of these. So current federal law requires that a drug be the subject of an approved marketing application before it's distributed across state lines. Because a sponsor probably will want to ship the drug to clinical investigators in many states, it needs to seek an exemption from that legal requirement. And an IND is the means through which they technically obtain that exemption from the FDA, which allows them to ship the product across state lines. This, of course, allows them to initiate clinical studies in humans. There are three main types: investigator-initiated INDs, which I think would be the most common in most situations where you're studying a drug to meet an approval standard. There are also emergency use INDs as well as treatment INDs. There are two categories. There are commercial INDs. These are the ones that are initiated by a sponsor who intends to eventually seek some sort of a labeling change or approval of a product related to the studies that are conducted under that IND. For studies that are being conducted more for academic or research purposes, there are also research INDs. All INDs must include preclinical data, manufacturing information, as well as clinical protocols and information on investigators that will allow the agency to evaluate whether the study that's proposed is reasonably safe to proceed. Not all studies of drugs need to be conducted under IND. There are exemptions for specific types of studies. So first, the product has to be lawfully marketed in the United States already. This cannot be a new product that is not already on the market. The study cannot be intended to support a new indication or a significant labeling change for the drug. The study itself cannot involve a route of administration, a dose, a patient population, or some other factor that would significantly increase the risk to participants in the study. And the investigation needs to be conducted in compliance with other regulations for instance, institutional review board, informed consent, and promotion. So what is the purpose of an IND? The IND provides the data needed for FDA to ensure the safety and rights of clinical study participants are maintained, and also to ensure the quality and adequacy of studies intended to provide evidence of the safety and effectiveness of medical products. After a sponsor submits an IND opening study protocol, the agency has 30 days to review the protocol, at which point the protocol will go into effect, unless the sponsor is notified earlier by FDA that the study may proceed or FDA issues a clinical hold. When the agency reviews these INDs or reviews any submission to an IND at any point in a development program, one lever we have if we have concerns about the study is to issue a clinical hold. This is an order that's issued by the FDA to delay a proposed clinical investigation or to suspend an investigation that is already ongoing. When we issue a clinical hold, no new subjects can be recruited or given the drug, and the patients in the study need to be taken off therapy unless they're permitted to continue by FDA in the interest of public patient safety. For instance, if stopping the drug abruptly would be harmful, there can be a plan to manage that in particular situations. Not all clinical holds are complete. There are partial clinical holds. For instance, if you have enough data to support lower doses but not higher doses, there may be a clinical hold on administering higher doses. Or for instance, if you have enough data to support studies in adults but not children, the studies could be allowed to proceed in adults while there's a clinical hold on studies in children. So what are the grounds for a clinical hold? For phase 1, 2, and 3 studies, the agency could find that there is really unreasonable and significant risk of illness or injury to participants in the study. There may not be enough information to understand the risk, so there may just be more information needed before a particular study or part of a study could move forward. The investigator brochure, which is intended to provide information to investigators on what is currently known about the drug, may be misleading, erroneous, or incomplete and need to be corrected to make sure that information on the drug is being accurately and completely portrayed to investigators who are enrolling patients in the study. The clinical investigators may not be qualified to conduct the investigation. And then lastly, if there is a disease that is life-threatening and affects both genders, but one gender, often women, are being excluded, that could be another ground for a clinical hold. For phase 2 and 3 studies, all of the above apply, but in this case, the agency can also put a study on clinical hold if the protocol is believed to be deficient in design to meet the objectives of the study. So moving on, once you have completed all of your studies under IND, you're ready for a marketing application. As I mentioned, there are two main types of marketing applications. One is a new drug application under the Food, Drug, and Cosmetic Act, and the other is a biologics license application under the Public Health Service Act. Both are a mechanism to provide the FDA with information that's needed to support the marketing approval of a new product, or to provide additional information to support a new indication or changes to labeling for an already approved product. Just to take a bit of a step back to a couple of basic definitions now that we're getting into new drug applications and biologics license applications. A drug is defined in the Food, Drug, and Cosmetic Act as articles recognized in the official United States Pharmacopoeia, official homeopathic pharmacopoeia of the United States, or the official national formulary. And they're articles that are intended to use in the diagnosis, cure, mitigation, treatment, or prevention of disease in man or other animals. And they're articles other than food intended to affect the structure or any function of the body of man or other animals. And then finally, they're articles intended for use as a component of something specified in A, B, or C. A biological product is a little bit different. It is defined in the Public Health Service Act as a virus, serum, toxin, antitoxin, vaccine, blood component, etc. Again, that's applicable to the prevention, treatment, or cure of a disease or condition in human beings. So all biological products would also meet the definition of a drug. So what do we receive in a marketing application, meaning an NDA or a BLA? First is chemistry, manufacturing, and controls information. So how is the product manufactured? How will it be packaged, et cetera? Non-clinical pharmacology and toxicology information. What is known about toxicities that were seen in preclinical studies or animal studies? Human pharmacokinetics and bioavailability studies. So what is known about the clinical pharmacology characteristics of the product? Of note, later in this course, you'll have lectures on all three of these topics from some of the experts in these areas. For anti-infectives, there's information on the microbiology of the product. And then, of course, there's clinical information from studies in humans, clinical trials, and other types of studies, statistical information, as well as information related to potential pediatric use of the product. We get samples, as well as information on product labeling. And then for participants in trials, we do get case report forms describing events that may have happened during the studies, as well as data sets where we can actually reproduce some of the analyses necessary to confirm the efficacy and safety findings of the study, as well as to conduct additional analyses if needed. And then finally, we receive legal information. So information from the applicant about patents, exclusivity, financial certifications, and so on. Product. So the key decisions before us when we get either an NDA or a BLA are whether the drug is safe and effective in its proposed uses, and whether the benefit of the drugs outweigh the risks of the drug, whether the labeling is appropriate and what it should contain, so providers who read the label understand how to use the drug and who it would be beneficial in. And then finally, whether the manufacturing methods used to maintain the drug's quality are adequate and that the methods preserve the drug's identity, strength, quality, and purity. And so at the end of the day, if we find all of these to be true, we will approve a drug and it will be legally marketed at that point. I'll move on to the challenge question. So the first challenge question is for a drug that is lawfully marketed in the United States, an investigational new drug application is never required to conduct a clinical investigation using the drug. Is this true or false? So this is false. Not only does the drug have to be lawfully marketed in the US, but to be exempt from IND requirements, the study can't be intended to support a new indication or significant labeling change. It can't involve a route of administration, dose, patient population, et cetera, that would significantly increase risk. And the investigation needs to be conducted in compliance with other regulations, such as IRB and informed consent requirements. So challenge question number 2, which of the following is not a reason for a clinical hold? A, the investigator brochure is incomplete. B, there's insufficient information to assess risk. Or C, the investigation of a drug for disease that manifests in childhood excludes children. Or D, clinical investigators are not qualified, which is not a reason for a clinical hold. So C is not a reason for a clinical hold. Just to briefly summarize my talk, FDA has been working to promote and protect public health for over 100 years. It's one of our oldest consumer protection agencies. The laws, regulations, and guidance provide a framework for the development and approval of drug and biologics and really frames the work that we do day to day at FDA. INDs are the mechanism through which FDA evaluates whether clinical investigations are reasonably safe to proceed, and NDAs and BLAs are the mechanism for our oversight of the US sale and marketing of drugs and biologics. Thank you very much for your attention. I will be back a bit later during the question and answer period, but now I will turn it over to Dr. Jim Smith.